Good afternoon, everyone. Today we are going to discuss about pharmaceutical emulsions. First, we uh, first we discuss what is emulsion. Emulsion is the biophasic dosage form which contains oil and water. Water means liquid and oil. Here you see clearly that an emulsion is a two-phase system consist of two incompletely miscible liquids. Please note that incompletely miscible liquids. Incompletely miscible means they are not mixed wellly. Okay, one of which is dispersed as a finite globules in the other means one phase other maybe it will be a liquid or it will be oil. They are from a finite globule when they are combined with each other. The particle size of the globules ranges from 0.25 to 25 micrometer. An emulsifying agent and mechanical energies are needed to join this phase. When you add oil and liquid mix together, then normally they are separate. Okay, they form a two separate layers. But when you use the emulsifying agent or mechanical energy like mechanical stirrers or other mechanical energies, they are going to form a emulsion, that is they form a globules and they are dispersed all over the phases. Firstly, we discuss what is the two types of phases. First is dispersion phase. Second is a continuous phase. Dispersed phase is a globules form, and continuous phase is a maximum phase available in this beaker. Okay. Then we are discussing what is the types of emulsions or classification of emulsions. First is the dispersed phase. On the basis of dispersed phase, second is particle size, and third mode of action. On the dispersed phase, there are classified in two types. That is, first is simple emulsion, second is multiple emulsion. In the simple emulsion, there is only oil in water types of emulsion and water in oil type of emulsion. You can see clearly that oil oblique O oblique W type or W oblique O type is the oil in water and water in oil type of emulsion. And second classification is multi emulsion. Multi emulsion is a different from the oil in water and water in oil, because multi emulsions contains three phases like oil oil or water oil water. We will discuss in the next part. Then second classification is the particle size. In the particle size, there is a micro emulsion and macro emulsion. It is a depend on the globule size present in the emulsions. Okay, micro emulsion and macro emulsions are being differ from the globule size. Second, and mode of action. Mode of action means how can we administer the emulsion in our body. First is the topical, second is rectal, third is the oral, and fourth parenteral. We will discuss in detail in the next part. Okay, there are four main types. First is the oil and water type. In the oil and water type, oil is a dispersed phase. Okay, oil is a dispersed means the oil is fully dispersed in the water. Dispersed phase. Water is dispersion medium preferred for internal use. This type of emulsion, oil in water type of emulsions, are preferred for the only internal use, non-greasy and easily removable from the skin. There is a non-greasy and easily removable because there is a maximum amount of water present in the oil and water type of emulsion. Okay, used externally to provide cooling effect. If you use this emulsion in externally, so it provide the cooling effect. Example, vanishing cream. Okay, the next is a water in oil type of emulsion. Water in oil type of emulsion. Water is dispersed phase and oil is dispersion medium. Okay, there is a difference between dispersed phase and dispersion medium. Dispersed phase is a less quantity. Okay, you can see on the screen that finite globules present in the big circle. These are the globules. Okay, in the oil in water type of emulsion, the yellow globules, they are yellow globules are present, that oil droplets, and big circle are present in the water. And in the water in oil type of emulsion, the small globules are present in act as water, and a big circle is the oil. Okay, greasy and non-water washable and used externally to prevent evaporation of moisture surface of the skin. greasy and not water washable. Okay, this is uh, this is the different from oil and water type of emulsion. Due to the maximum amount of oil present in the water in oil type of emulsion, they are greasy. Okay, and prevent evaporation of moisture skin. Whenever you apply on your skin, they hold the moisture content in your skin, and they are resist to evaporate the moisture content. Example is a cold cream. Next is a multi-emulsion. In the multiple emulsion, dispersed phase contains smaller droplets that have the same composition as the external phase. We are earlier discussed in the classification the multi-emulsion. Multi-emulsion, you can see clearly that there is a two diagrams which are appeared on your screen. One is water in oil in water type of emulsion, and second is oil in water in oil type of emulsion. There is a maximum white colored diagram shows that water in oil in water. Firstly, you clarify that the white color indicates the water and blue color indicates the oil. Okay, whenever you see the diagrams, there is a two diagram. Okay, there is a first diagram is water in oil in water. Okay, they have a maximum amount of white color, and second is oil in water in oil that contain a maximum amount of blue color, which indicates the oil is continuous phase and water is a dispersed phase. Okay, then we are going move forwards. Water in oil in water system and oil phase separates internal and external aqueous phase. Okay, the medium globules are used to separate the inner phase and outer phase. Oil in water in oil in a system and water phase separates internal and external oil phase. Then this is the fourth type of emulsion that's based on the particle size. In the particle size, 0.2 to 50 micrometer globule size act as a macro emulsions. Okay, 0.2 to 50 micrometer globule size act as a macro emulsion. 0.01 to 0.2 micrometer globule size act as a micro emulsion. Please clarify that the sizes of the micro emulsion or macro emulsion. 0.01 to 0.2 micrometer is a global size for the micro emulsion, and 0.2 to 50 micrometer act as a global size for the macro emulsion. Okay, then we will move forward. Formulation of emulsion. There are three main ingredients which are responsible for the stable emulsion. We use oil, water, as well as the other ingredient which are used to the keep the emulsion stable. First is the emulsifiers. Selection of emulsifying agent EA. An emulsifying agent is a material that enhance the stability of emulsion. Clarify that it is used to enhance the stability of an emulsion, that is prevention of coalescence and reducing creaming. What is coalescence? Now, coalescence is when the globules are get together and form a single globule, a single big globule. That is called as a coalescence. There is a two terms: coalescence and flocculation. Coalescence is when the globules are get interact with each other and form a big globule, and flocculation is a small globules are get together, but they did not combine with each other and not form a big globule. Okay. The ideal emulsifying agent is a colorless. Note that the ideal emulsifying agent's characteristics: first is a colorless, second odorless, third is tasteless, non-toxic, non-irritant. Okay, and able to produce stable emulsion at low concentration. Now, how we form a how emulsifying agent form a stable emulsion? Emulsifiers act as a surfactant, that is they reduce the surface tension between two layers. Okay, one is aqueous and oil. Okay, they are reduce the surface tension between them. Examples of emulsifying agents are enlisted there: carbohydrate materials like acacia, tragacanth, agar, pectin for the oil in water emulsion, and protein substances like gelatin, egg yolk, casein for the oil in water emulsion. Please note that carbohydrate material for oil in water emulsion and protein substances for oil in water emulsions contains gelatin, egg yolk, and casein. Second major ingredient we used in the emulsion is preservatives. Preservative is majorly used for the inhibition of growth of microbes. Okay, inhibition of growth of microbes. There are several examples like benzoic acid, salicylic acid, benzalkonium chloride. Okay, their major question is why we used preservatives. Everyone is using the preservatives, but why we use preservatives? The number of ingredient in emulsion supports the growth of microorganisms which result in change in color, odor, taste of emulsion. Whenever we don't use the preservative, there are maybe chances to growth of organisms which are results in the color or odor or taste of emulsion changes. Okay, preservative used in emulsion, they must be there are certain criteria for the using of preservatives. They are must be less toxic, chemically stable, reasonable cost, acceptable odor, taste and color, effective against fungi, yeast, bacteria, stable to heat and storage. Okay, they are the when we use emulsions for the internal use, then there must be a less toxic. Okay, second is chemical stable. If the emulsion is not chemical stable, they form a cracking on the emulsion and they separate the phases. Second is the reasonable cost. Uh, whenever we uh, whenever we produce the emulsion in the industry, there are more amount of preservatives we require, so it must be a reasonable cost. Second is an acceptable odor, taste and color. When we administered orally emulsion, when we are administered orally, there must be a better odor, taste and color. Okay, effect the main use of the preservatives is effective against fungi, yeast and bacteria. If it is not effective, then it is a worthless. Okay, so finally the stable to heat and storage. uh, there are uh, there are many formulations which are not stable at the room temperature also. Okay, so other than the ingredient, the preservatives must be stable at heat and storage. Next is the third, antioxidant. Antioxidant inhibited oxidation of the emulsion. Okay, antioxidant are used to inhibit oxidation of the emulsion. What is the oxidation? Okay, oxidation is a chemical reaction that can produce a free radicals, thereby leading to a chain reaction. Please remember, oxidation is a chemical reaction that can produce free radical and thereby leading to the chain reaction that may damage the cells of organisms or the uh, uh, it becomes the unstable emulsion. Okay, antioxidants are compounds that inhibit oxidation. Example: gallic acid, ascorbic acid, etc. Ideal properties of antioxidant. There are several criteria for the antioxidant. First is non-toxic, non-irritant, effective at low concentration under the expected conditions of the storage and use. Remember, effective at a low concentration. Uh, that is, we used a minimum quantity of the antioxidant and it is effective largely. Okay, low concentration under the expected condition of storage and use. Soluble in the medium and stable. It must be soluble. Okay, if it is not soluble, then it forms the it settle down on the emulsion and that is not act as an antioxidant. Antioxidants for use in oral preparation should also be odorless and tasteless. It must be odorless and tasteless. Okay. Next is the preparation of emulsion. How we prepare the emulsion? There are two main methods. First is a trituration method and second is a bottle method. In the trituration method, there are another two methods. First is a dry gum method and second is a wet gum method. What is the difference between them? In the dry gum method, in this method, the oil is first triturated with gum with a little amount of water to form a primary emulsion. The trituration is continued till a characteristic clicking sound in the heard. Okay, whenever you click the mouse, that like sound it must perform. Clicking sound is heard and a thick white cream is formed. Once the primary emulsion is formed, the remaining quantity of water is slowly added to the form the final emulsion. Okay, whenever the oil and the gum mixed well, then you can add the remaining water and form the final formulation. Okay, second is a wet gum method. In the wet gum method, as the name implies, is in the method, first gum and water are saturated together to form a mucilage. First gum and water is saturated together to form a mucilage. The required quantity of oil is then added gradually. Okay, whenever you require water, then you add the small quantity of water and uh, add and saturate gradually in small portions with a through the saturation to form the primary emulsion. Then you have to form a primary emulsion. Once the primary emulsion has been formed, remaining quantity of water is added to make the final emulsion. Okay, whenever you form a primary emulsion, okay, whenever you form primary emulsion, then the remaining quantity of water you have to add in the emulsion and make a final emulsion. This method consists of 4 as to 2 as to 1 formula. Okay, 4 part of oil, 2 part of oil, water, and 1 part of gum. Remember, 4 as to 2 as to 1. The next method is a bottle method. In the bottle method, it is extemporaneous preparation for volatile oil or oil with low viscosity. It is especially used for the having the volatile oil or oil with low viscosity. Firstly, we have to add gum plus oil in the dry bottle. Then we have to shake it well. Then water volume equal to the oil. You have to add the water volume equal to the oil is added in a portion with vigorous shaking to form a primary emulsion. Okay, when firstly you took a gum and oil, then how many quantity of oil you taken, same quantity of water you have to take and shake it vigorously and form a primary emulsion. Then remaining quantity of water is added to make the final emulsion. Remaining quantity of water, that is, firstly you took a 10 ml of oil, then you have to also take a 10 ml of water, but the final quantity of water as a 50 ml, then remaining quantity of 40 ml water you have to take, you have to add in that bottle and shake it. Okay, then you form the emulsion. Then there is a several identification test of emulsion. First is the dilution test. The test is based on the solubility of the external phase of emulsion. Okay, in the dilution test, oil in water emulsion can be diluted with water. The major continuous phase is used for the dilution. Okay, always remember, if the water is the continuous phase and oil is the dispersed phase, you have to add a water. Okay, then if the water is a dispersed phase and oil is a continuous phase, it can be diluted with the oil. Okay, there is a three diagrams shown in your screen. That is, first is oil in water emulsion. Then if you oil in water emulsion diluted with water, okay, there is an arrow diluted with water. Then what happens? No phase change, there is a no any phase is changed. Oil in water emulsions become diluted or no any phase is changed. Okay, then again diluted with oil. What happens if the diluted with oil, there is a two different phases are appeared, that is oil and water. Okay, then final picture shows that oil and water emulsion forms a two different phases, that is a phase change are occurred. Then second is a dye test. In the dye test, when emulsion mixed with amaranth dye and observe the under microscope, if the emulsion mixed with amaranth dye is a dye which are used to color the oil. Okay, sorry, to color the water. Okay, there is a first slide and there is a two slides are shown in your screen. First is a oil in water emulsion, and second is oil in water emulsion. Okay, water and oil emulsion. First slide, that is maximum red color, shows that the maximum color which are shown in the first image, that is for the water. Okay, oil and water emulsions. Oil is a white globules and water is a become a reddish part. Okay, in the second slide, water and oil emulsion, the water forms a finite globules and oil is a major part, that is it shows a maximum white color. Okay, if continuous phase appears red, then it means emulsion oil in water type. If continuous phase appears red, then it means a oil in water type. If the scatters of globules appears red and continuous phase is colorless, then it water in oil type. Okay, remember, first is a dilution test, where we dilute the emulsion with the water or oil and see the changes, that is they form a phase separation or not. Second is the dye tests, where we use the amaranth dye, and the amaranth dye colors the water, and it shows the reddish color when a maximum reddish color when the oil and water type of emulsion, and it shows dotty structure, that is shown in the second images, that is for water and oil emulsion. Second is a second view. Second, we discuss what is the good emulsion versus bad emulsion. There is a five beakers. The center beaker is for the good emulsion, and there is a problems occurred in the bad emulsion. Is first is coalescence, second is flocculation, third is creaming, and the fourth is a breaking. Okay, coalescence. I earlier discussed what is coalescence. Coalescence when the globules are come together and form a big globule. Okay, that is shown in the picture. The globules are come together and form a big globule, that form a coalescence. Due to this, maybe phasor become separated, and emulsion are become not stable, unstable. Okay, second is a flocculation. Flocculation means the globules are being together and not to form a big globule, but they are form an aggregation like globules. And third is a creaming. Creaming means there is a foamy structure appeared on the surface of the emulsion. Okay, foamy structure are appeared, that is oil are become a cream, and it appears on the surface of the emulsion. And finally is a breaking. Breaking is a fully separation of phases, that is oil and water. Okay, these four are the problems occurred during the formation of emulsions. That's why we use the emulsifier agents that are used for the uh, well dispersed globule formation. Okay, thank you.